Hello, my name is Royce Cumming, and welcome to my presentation on Phasmatodea camouflage and its phytochemical basis. Now, please bear with me today, as I am actually an entomologist and evolutionary biologist, and have only begun to dabble in the world of carotenoids, thanks to Dr. Eleanor Wurtzel at Lehman College in New York. For those of you who are not familiar with phasmids, they are the stick and leaf insects which can be found around the world and come in a beautiful array of colors, forms, and sizes. With some as long as a human arm, and others able to fit on the tip of your finger. But what phasmids are best known for is their impressive camouflage, masquerading as moss, lichen, leaves, and sticks in order to seamlessly blend right into their environment to avoid being eaten by predators. And not only have they perfected their morphology to match their arboreal habitat, but their behaviors have evolved to really drive home their masquerade. Even their textures and choice in habitat are fine-tuned for perfect camouflage. My particular background has primarily been working on the evolutionary history of the Phyliidae, or walking leaves, a flat group of phasmids which are arguably some of the best leaf-mimicking animals out there. And to show you just what I mean, I'm going to give you a couple seconds to pretend you're a hungry bird looking for an easy meal in the canopy. How many leaf insects can you see? Now, to be fair, I only have enough time to let you look for a moment, so I will reveal them in 3, 2, 1. Did you find all 9 leaf insects hiding in the canopy? Pretty tough, even for a trained entomologist. Now, as far as camouflage is concerned, multiple studies have reviewed the different elements that make up camouflage, such as coloration, light reflectance, movement, texture, etc., that make up a believable act in order to fool a predator. And what has repeatedly been found is that the perfect matching of coloration was more influential to avoid detection than other aspects for identifying prey. When you think about it, it makes sense. In a world full of variable shapes and sizes, and especially in a canopy full of movement, if you have an adaptation that helps you perfectly match the colors around you, you are less likely to be spotted than another that developed the wrong coloration for this particular habitat and ends up getting eaten. By now, I'm sure you are thinking, Royce, this is all cool, but what does it have to do with carotenoids? Well, as you all know, carotenoids have many functions in animals, including vision, pheromones, vitamins, antioxidants. But importantly for insects, they're also commonly used for coloration and are modified in different ways according to the insect's particular needs. Therefore, our central hypothesis is that the differing ability of phasmids to sequester and modify carotenoids is linked to phasmid morphology and ecology evolution. One ecological aspect which is closely tied with coloration are the early stages of life, as many phasmid eggs, as seen on the left, are mimicking seeds, and many species just fling the eggs to the ground, which, when they hatch, the nymphs are typically brown, red, or black, to blend into the dead leaves on the forest floor. This is an issue, of course, because as soon as the nymphs walk into the green-dominated canopy, they stick out as clearly the wrong color. Amazingly, the phasmids have found a way to work around this issue, as phasmids have evolved the ability to change their colors to ensure they always blend right into the environment and maintain that perfect camouflage. This ability to modify their coloration is actually pretty rare in the insect world, as many insect colors are on or within the outer cuticle of the exoskeleton and are permanent, unable to be changed. Whereas phasmid coloration is within living cells below the cuticle in the epidermis, and therefore can be changed as the contents of the cells are changed. The ability to change colors facilitates important adaptations for these masters of camouflage, such as the ability to change their coloration to match the environment, the host plants they are living on, adjust to the seasons, or rapidly change as their lifestyle changes, like you saw with the freshly hatched nymph going from red on the ground to green in the canopy. Unfortunately, there has not been a great deal of work done with phasmid carotenoids over the years, but the few species which have been reviewed thus far have had some overlap and some novel carotenoids recovered, with many appearing to be derivatives of beta carotene. Unfortunately, few species have been reviewed so far, with many somewhat closely related, not yet spanning widely across the phasmid family tree. Although these works have helped to pave the way for our proposed study, there are a number of shortcomings. One issue is that they did not control for host plant, so they did not know if the insects were simply sequestering directly from their diets or actually modifying the functional groups. Another is often their results were recorded as presence-absence, not as quantities, so the ratios of different carotenoids in the insects are unknown, which, for our coloration work, the ratio of different carotenoids will influence their overall coloration. And finally, for most studies, the entire insects were sampled, so they were reviewing more than just is present in the exoskeleton. Thankfully, with phasmids so popular as pets and in live insect exhibits like the Long Island Aquarium, who we are partnered with, we now have easy access to an evolutionarily significant assortment of species, which can be characterized to help keep track of exactly what carotenoids they were feeding on throughout their development in a controlled setting. At present, we are still perfecting our extraction and separation techniques, but we have so far had promising results from two distantly related species, both bright green in color, but with notably different carotenoids sequestered. Some, such as our tentatively identified beta cryptoxanthin, was exclusively within the insects and present in both species, but not present within the host plants they were feeding on. Whereas others, such as beta carotene, were recovered from all three samples as relatively prominent. Once we are satisfied with our techniques and confident that we are recovering all carotenoids present within the host plants and the insects, we will be moving on to a wider diversity of species across the phasmid family tree. Now in the age of genomics, their evolutionary history is being revealed with greater clarity, and as we find similarities and differences in how phasmids modify carotenoids, we can use this trait to reveal overall evolutionary patterns and associated traits. So hopefully by the next conference, I'll have a great deal more to share with you all, so please stay tuned. And of course, a big thank you to Jeff Petraka, George Britton, and Kaiji Zhu. Thank you.